Thank you for joining today. Just as as always, I'll just give a brief introduction to all the new members about IC Impacts and UBC Future Waters. So IC Impacts is a international and national center of excellence hosted here at UBC. Uh, it's a group of more than 1,000 graduate students, and it's one of the initiatives that we have started of hosting online webinars. Um, Similarly, UBC Future Waters is a group of UBC Research Excellence Cluster. Um, Again, it has a big network of students, and we or the organizing team is a part of it as well. So just to brief you, the organizing committee, you can see me, there's Jaskurin from McGill, Abhishek from UBC West, Feria and Carl all have been playing a leading role in organizing these WESTalks, and I guess you would have seen them quite often in previous talks as well. So just to brief you of the talks that we have had so far and what's coming. So this is probably this is our sixth WESTalk. Last week was a part of our annual general research conference. Today we have Dr. Elimelech from Yale University, and we have these webinars going on for quite some time now. So it will probably happen until October, and you can see the upcoming talks for the month of September and October as well. So if you guys are interested, please join our Eventbrite page or register at the Eventbrite page. And if you're a student and you want to get involved in any way, just send us a message. We are open to anyone from any part of the country or any part of the world, actually. With that, I would like to introduce today's speaker. I won't introduce Dr. Jongo Lee, who is a professor within the civil engineering department here at the University of British Columbia, would be introducing a speaker. He has been quite helpful right from the beginning in organizing these WESTalks and has worked with this Dr. Elimelech for quite a few times. So, Dr. Lee, could you kindly introduce today's speaker, please?
Sure, yeah. Thanks for her. Yeah, hello everyone. My name is Jong-Ho Lee. I'm an assistant professor in the Department of Civil Engineering at UBC. I'm pleased to introduce today's guest speaker, Dr. Menachem Elimelech. So Dr. Elimelech is a Roberto Goizueta professor at the Department of Chemical and Environmental Engineering at Yale University. His research focuses on membrane-based technologies at the water energy nexus, materials for next generation desalination and water purification membranes, and environmental applications of nanomaterials. But Dr. Elimelech was the recipient of numerous awards in recognition of his research contributions. There are too many to list, so I just want to mention a couple of them. He received the 2005 Clark Prize for Excellence in Water Research, election to the U.S. National Academy of Engineering in 2006, and ENI Prize, ENI Prize for Protection of the Environment in 2015. Just to mention that this ENI Prize is very highly prestigious award in the field of energy, and many people call this prize even as Nobel Prize for Energy. Also, as you can notice from his H index of 155 and total citations almost 100,000, Dr. Elimelech is highly cited researcher in chemistry, environment, and ecology. Dr. Elimelech is also well known as an exceptional educator and mentor. He has advised 41 PhD students and 36 postdoctoral researchers, and more than 50 of them are now in academic positions, and others hold also leading positions in both in academia and industry. I guess I'm one of the most successful former postdocs of his because I got a job at UBC. I'm just kidding. In recognition of his excellence in teaching and mentoring, he received the W.M. Keck Foundation Engineering Teaching Excellence Award in 1994, the Yale University Graduate Mentoring Award in 2004, and the Yale University Postdoctoral Mentoring Prize in 2012. With that, I'd like to hand over the floor to Manny. Manny, the floor is yours. Thanks.
Okay.
Okay, thank you, John Gol, for this kind introduction, and I'm very pleased to present a seminar in this series. The title of my presentation will be Next Generation Desalination Membranes, and I will try to tell you where we are really now. And it will be more a high-level presentation with some technical detail, but I would like you mostly to get the big picture, where these membranes are now. So let's start with a background information that is probably most of you familiar. Water scarcity is a global concern. 4 billion people face severe water scarcity more than, let's see, I cannot, just a
Many
second.
you have to share this. Start sharing the screen again.
Oh, okay.
Yeah, that works.
Okay, you see me now? I don't know
Yeah,
what happened, but yeah, okay. So anyway, so I'll talk about global water scarcity is a major concern. You can see here 4 million people face severe water scarcity more than 1 month a year, 500 million all year round. And when you look at the maps for water scarcity, it can be quite confusing. Let's look at this map, for example. Here you see the water scarcity for each country. And you see if you have very yellow, it means very little, like Canada, and United States is more like medium to moderate to high, moderate stress. And these kind of maps are based on one country, but they're quite deceiving because some countries are quite large. So what we really need to see, we need to look at the regional water scarcity, as you see in this map for the United States. And when you look at this regional water scarcity, you see really quite severe water scarcity in the south west, as you see here. And although there is a lot of water in the East Coast, I mean, it's not practical to transfer water all the way from the water-rich states to the water-deprived states, because it needs a lot of energy. Now, we all heard about Cape Town, that they almost closed the turn of the water to the city because the reservoirs were very, very low level. And eventually, weeks later, the BBC wrote an article about other 11 cities that can run out of water. And you can see in this list cities that you will never think about it because some of them are really close to some major rivers. And the reason is not only that there is lack of water, but many times it can be pollution or very bad water management. The combination of all of these pollution, water management may result in some water scarcity in one of these 11 cities that you see here. And I emphasize water management because the city of Chennai, formerly Madras in India, did not do the right water management, and it did last year. They practically ran out of water. If they did the right management and they were thinking in advance, like building desalination plants, and I'm going to describe later, probably they would have avoided this crisis. So this brings me to the general talk of my subject of my talk, desalination. And desalination can be used as a measure to augment water supply. We can get a certain amount of water from the hydrological cycle, like from rivers, lakes, open reservoirs, but it's not enough. And the only way to get more water is through using engineering. We are all engineers. We can either desalinate seawater or reuse municipal wastewater. So this is what I would like to talk here. And let's start with desalination. So reverse osmosis is the gold standard for desalination. So I think my camera, I have some issues with my camera. Okay, so reverse osmosis is the gold standard for desalination. I'm just checking if my camera is okay. I think there is some problem with my camera. Let's see if I can fix it. So just a second. Okay, now let's see, where is my, okay, here. Oh, just a second, sorry for all this. Okay, I think I fixed everything. Okay, so reverse osmosis is the gold standard for desalination. And this is a typical reverse osmosis plant. You have intake of water, pre-treatment, as you see here. Reverse osmosis, the stage that removes the salt, some post-treatment to some minerals and other treatments, and then discharge to the ocean. Okay, so let's give you some example of some major desalination projects, and I will go to Israel, Israel really has more probably desalination plants per capita than any place in the world. And these are 5 major desalination plants that were built along the Mediterranean coast over a distance of less than 120 kilometers, as you see here. Israel is a very, very small country. And this is the names and the capacities of each of these desalination plants. For example, one of them, Sorek, is 110 million gallons per day. And just to put everything in perspective, California, after 12 years of planning, built 1 desalination plant. And as you see here, the desalination plant in Carlsbad, it's only 50 million gallons per day, less than the smallest one in Israel. So just to put everything in perspective. Now, another way to augment water supply is through desalination of wastewater. Again, when you talk about wastewater, you say, why desalination? But indeed, there is some desalination step there. This is a typical wastewater reclamation plant, advanced wastewater reclamation plant. You take the wastewater, and rather than discharge it to the ocean, you go through some advanced treatment, pre-treatment by microfiltration, reverse osmosis to remove salt because wastewater is salty above the limit for drinking water supply, then advanced oxidation, and in principle, you can produce water of potable water quality. And one great example is Orange County, California, in which this plant in Orange County, the groundwater water replenishment system treat 100 million gallons per day by the scheme that I showed you earlier. And then they inject the water in the groundwater, and the groundwater is used as water supply for Orange County, California. So this is what we call an indirect potable use. The water put in the groundwater, there is some further treatment by Mother Nature, by natural processes, and when you pump the water a few kilometers from the site, you can drink water that is also including wastewater. Now, in all these processes, membranes play a very important role. And I would like to focus on the desalination membrane for the scheme that I just showed you for advanced wastewater treatment plant. This is the reverse osmosis stage that does the desalination, and I will talk about the membranes that use there. Similarly, for the seawater desalination plant, the main stage is the reverse osmosis, as you see here. And this reverse osmosis also utilizing desalination plants that are the subject, desalination membranes, which are the subject of my talk. Now, what are these desalination membranes? So the gold standards for desalination are the TFC polyamide thin film composite membrane. Thin film composite polyamide membranes. And they are the gold standard since 1980. The first patent and the invention of this membrane was done in the 1980. And since then, 40 years, no one was able to get a membrane better than this membrane, despite all the efforts in industry and scientific community. And a few words about this membrane. It's formed by interfacial polymerization of 2 monomers on an interface of aqueous solution and the organic solvent. And then you can form a very, very thin selective layer of only a couple hundred nanometer. And this selective layer gives you the right permeability and salt rejection that needed for seawater desalination. So what I would like now to get to my talk and talk about the evolution of next generation membranes. Again, there were many attempts to have a membrane better than this thin film composite membrane, which is 40 years old. It's very old membrane in principle. And I would like to go over the research, and this will be sort of like, will frame my lecture. I will first talk about ultra-high flux membrane or ultra-high permeability. This was a very active area of research between 2006 up to 2014, and they realized that they were not in the right direction despite all these fancy papers. Then the research moved to a water salt selectivity, how to create a membrane with very, very high salt rejection, which is better than the current membrane. And you will see why we need this high salt rejection. And lastly, what I will talk in the end is what I think the future, and now you're starting to see some ongoing work. This is about membranes for ion-ion selectivity that can be used for different applications, including desalination of brackish and wastewater. So as I told you, this will frame my lecture. I will talk about water permeability, water source selectivity, and ion-ion selectivity. So let's start with the first one, which will be water permeability. So as I told you earlier, from 2004 until 6 until 12, there was a lot of research on high flux membrane, and they promised a quantum leap in desalination. They promised that such membrane will reduce the energy consumption to such a way that you'll be almost, you need no energy to desalinate. So again, and they were talking about membranes like carbon nanotube that you see here on the left, that they can have very high water flux because of their unique properties, hydrophobicity, and very smooth surface. There were some papers about aquaporin-based membrane that also will have high selectivity. And then also about graphene. Graphene, again, is highly permeable. And all these research promise really, if you can get very high permeability, we can reduce the energy of desalination, and then it will be really a very simple process that everyone can use, including even in Vancouver. So, and again, there were plenty of paper on this, and again, all these high impact journals, Science, Nature, I think that many of you, wish to publish, and me also, and so on. And we thought about it, and we did some analysis. And again, we published this paper, we analyzed a full-scale modeling of the energy consumption as a function of the water permeability. And I will show you here some data that we presented it recently in another format, of data of desalination of different kind of water, brackish, and then seawater from the Tampa Bay, typical seawater, and high salinity seawater from the Arabian Sea. This described the energy consumption in kilowatt hour per cubic meter. This described the water permeability. And again, the membranes, the high flux membrane, were supposed to increase the water permeability to very, very high, above 100. Now, for seawater desalination membranes, this is the current permeability of membranes, between 2 and 3 these units, LMH per bar, and these are for brackish water. So when you look at this analysis, you see that anything above the current permeability of the membranes, you see that the energy efficiency will improve by a few percent, 1 or 2% at most. Now, it was true for the older membranes before 1980, before we had the thin film composite membranes. The permeabilities of the cellulose-based membrane were much less. They were about 0.1, which is not even in this curve. And for such membranes, again, we needed high permeability. But once you got the polyamide membrane, the permeability is right. And again, it took a lot. We published it in several papers, and eventually people now realize and move to some other kind of research. So again, to summarize this part, again, there is a critical need. Again, there is no need for high permeability, but what we are trying to push, that there is a critical need for high selectivity. And high selectivity, it means high water transport compared to salt, or in simple word, ultra high salt rejection. So again, we need high selectivity. We need to move along this direction in these typical curves that show selectivity and permeability of membranes, and not high permeability, as I hope I convinced you based on the previous data. Now, I did not explain the mechanism, but again, when you go to the current permeability membrane, when you go to this salinity and 50% recovery, what really controls the energy consumption is really the osmotic pressure rather than the membrane resistance. So this brings me now to the second topic, and here I will provide more details about water source selectivity and why we need high water source selectivity or high salt rejection. Let's have an example in seawater desalination. In seawater, there is boron, which in a typical pH of seawater, it's an boric acid, as you see here. And those of you who know membranes, if you have a neutrally charged solute of low molecular weight like boron, it will not be rejected well by the membrane. So if we have membrane with very high salt rejection, we can reject boron, because boron can be detrimental to agriculture, and you need some additional reverse osmosis stages to meet the regulation for agriculture, like the case in Israel. Let's go to wastewater. We have many disinfection byproducts in wastewater, and they are neutrally charged, as you see here, like NDMA or some other toxic contaminants, and they will not be removed well by reverse osmosis, so you will need a more extensive advanced oxidation to oxidize them. So we need membranes with high selectivity or ultra high salt rejection, and this brings us here to some potential membranes that we discussed in a recent article that you see here. Again, it's not all recent anymore, but I will focus here on two kinds of membranes. One of them aquaporin based, and the other one synthetic channels. And I would like now to show the work of Jay Werber, which was a PhD student in my lab. He's now doing a postdoc, and he accepted the position at University of Toronto. Will start next year, so you'll be a colleague to many of you in Canada. And the current biomimetic membrane, the way you envision them and the way all the experiments done, that you will have this selective channel in what we call a fluid-like either lipid or amphiphilic black polymer bilayer. Lipid bilayer are not practical. Again, they are used in biological cells. They are not practical for engineered system. Most likely there will be some black polymer that you create this bilayer, and you will insert these membranes there. So we decided to have the following analysis. Suppose, again, and this will be very, very, very challenging. Suppose that you incorporate these channels into this bilayer with no defects, zero defects, which is, again, a daunting challenge. And then if we do it, what will be the limits of this membrane? What will be the performance limit of this membrane? What's the best we can get? And this is the analysis that we have done. And first, we take these various synthetic channels, either carbon nanotubes, aquaporin, or synthetic channels. We know what's the single channel permeability. And we said, what will be the areal density? How many of them we need to incorporate into the membrane to achieve the current selectivity of polyamide membrane, which is good, and we don't need more than it. And you see that what we can, the values, we see only, we need a very, very small percentage. For carbon nanotube, less than 1%, and for the least permeable channel, we need only 8%, which means 8% of the biomimetic membrane will be at most channels, and all the rest, 92 to 99, will be this lipid or black polymer bilayer. So it makes sense to study the performance of this bilayer. And this is what we have done. So again, the conclusion that the solute permeation will be determined by the surrounding bilayer because it's over 90% of the area. So what we do, we create vesicles. It's very common in this field. Soon you'll see that it's difficult and it's almost impossible to create flat membranes the way we know. And we can measure the permeability of these vesicles to different solutes, salt, water. And once we get this permeability, we can project what will be the permeability of current membranes. If you put them in a flat membrane, as I showed you in this diagram here. And we, again, I will not spend a lot of time here, but again, we can create this polymer, a black polymer bilayer. Again, this is an example of a polybutane with polyethylene oxide, PBPEO. Again, the color that you see here match the hydrophobic and hydrophilic. We were able to create it. This is a cryo-TEM of such vesicle. You can also get tri-black copolymer. And these are the colors that show you the hydrophobic, hydrophilic. This is the hydrophobic part, hydrophilic. And indeed, we were able to create such vesicles. Then we developed a method by stop flow kinetic and fluorescence quenching. I will not describe it here. Again, I was told that this talk should be between 30 and 40 minutes. And we were able to measure the permeability of a range of solutes and also water. And I will show you some examples. This is the permeability of a wide range of solutes that you see here, either diols, polyols, carboxylic acid. And we measured we present their permeability as a function of their octanol water partitioning coefficient, KOW, which tell you how hydrophobic is the compound. And we see beautiful correlation through this black copolymer of this, amphiphilic black copolymer. We see beautiful correlation of the permeability to the octanol water partitioning coefficient. I will not show you some data here, but we tried to correlate it to the size of the molecule, and we did not see any correlation. So it tells you, as we know from the biophysical literature, that the permeability through the black copolymer is solubility dependent. The solute partition into this black copolymer and then diffuses. This is what they call solubility dependent. And if you look at the values here of the permeability, we see that the PBPO has much lower permeability to this solute, which will make it a better candidate for desalination membranes. So now that we developed this method to quantify the permeability of this black copolymer that's supposed to be between 92 and 99% of the area of the membrane, let's see what kind of performance we can project for these membranes. So let's see, suppose we incorporate these channels defect-free, which is very difficult, if not impossible. So if we can do it, if you look at sodium chloride is very hydrophilic, so it will not partition into this black copolymer and will not go through this black copolymer. And here is an example for aquaporin, about 2 to 5% will give you the right permeability. This is the performance of this membrane, the biomimetic membranes. You see the perm selectivity is 10 to the 12, which is equivalent to 99.9999, 5 nines, salt rejection. And this is the performance of current membrane. You see below here, which is a perm selectivity of about 10 to the 4 to 10 to the 5. So the conclusion for seawater desalination, I mean, in terms of sodium chloride, you'll be quite good. Also for boron, such membranes, let's take the aquaporin Z with the PBPO, which is the best black copolymer. You see we can get perm selectivity of 10 to the 5, which will be close to 98, 99% rejection, compared to the current membranes that you see at the bottom, which is very low rejection. So the immediate conclusion here, that defect-free biomimetic membranes will be very good for seawater desalination, and not surprising. Again, we need to remove salt. Salt is very hydrophilic. It will not go through this black copolymer layer because it's hydrophilic, and these aquaporin channels will remove it quite well. Now let's go to wastewater application. We apply these membranes also for wastewater, and let's see how the performance will be for wastewater. And here it's a table, a graph that I show you a wide range of what we call micropollutants. A lot of pollutants which are low molecular weight, typically hydrophobic, as you see from the KOW, anything with positive log KOW, which means hydrophobic. And this is the molecular weight of these compounds. And let's see what will be the performance of such membranes. But before going to the aquaporin membranes, let's show you the performance of the current thin film composite reverse osmosis membrane for these micropollutants. These are observed rejection experiments versus molecular weight of these organic molecules that I showed you earlier. We see, as expected, that anything above 100 gram per mole molecular weight, we get high rejection. And anything less than 100 gram per mole, we get low rejection, which when we project it here, we see that most of the micropollutants have high molecular weight, so they will be rejected quite well, except, as I told you earlier, the low molecular weight, like 1,4-dioxane, NDMA, THMs. So this is for the reverse osmosis membrane. Now, what will happen if we will create our aquaporin-based membrane with the best black copolymer PBPEO and 3% coverage of aquaporin Z, which will give you very good permeability. And let's see, this is the perm selectivity, which again, also the rejection, as you go up, the rejection go high. So we see that when you have negative low KOW, which means when it's hydrophilic, we get high rejection. And when it's hydrophobic, positive low KOW, we get low rejection. And when we project it here, we see it really, it's really a major problem. All the contaminant in wastewater are hydrophobic, most of them. So they will go very easily through this membrane because the black, although the aquaporin will reject them, this black copolymer will not be able to block them because they will partition into the black copolymer and diffuse by the solution diffusion mechanism. So the conclusion here, defect-free biomimetic membranes, which again, defect-free will be very challenging, will perform poorly for wastewater reclamation. And again, there is a lot of push now in many places to go to wastewater reuse, like the push in California, rather than seawater desalination, which is environmentally, it will be more beneficial than just seawater desalination. I would like to say a few words. Scaling or upscaling these biomimetic membranes will be a daunting challenge. And when I use the word daunting challenge, I mean, it's almost for me to say impossible. There has been more than 1 decade research on these biomimetic membranes, and all the research was done on the vesicle level stage. Any attempt to make even a very small flat planar membrane failed. So this tells us, I mean, the failures of transition from the vesicle format to very small scale laboratory coupon, again, the one that are few centimeters square, suggests that scaling up to industrial level will be a daunting challenge. And again, not only that there will be daunting challenge because of the defect, but also the viability of this aquaporin, or the synthetic one, the performance for anti-fouling and so on. So this will be very challenging. It's very cool for research. And again, we have done also some research about it, but I'm very pessimistic about this kind of things for real world application. Now, again, we go to this, illustration, and we heard a lot about the graphene membranes. Again, we heard about them, that it will be, again, the next desalination membranes. And I would like just to say a few words about it. We have done some analysis, and to tell you what we think about it. So again, these 2D materials were proposed for desalination membrane. They propose if you have either graphene oxide or molybdenum disulfate, and if you put them as a laminates, and again, it was pushed by a lot of research from the University of Manchester, by the Graphene Institute, they promise a quantum leap in desalination. And again, these are the same thing, Sci, Nature, about graphene oxide, and about molybdenum and other 2-dimensional materials. Again, here, I mean, still you see Nature and all of these. And what they suggested, that how these membranes will work, if you create these laminates, the water can pass between these laminates if you have the spacing at about 0.5 nanometer. And the 0.5 nanometer will block the salt, and you will get membrane with complete rejection. And again, the way they create this membrane, usually you suspend this graphene oxide and you do vacuum filtration. And we were puzzled how something like this can work. And we're looking at the literature, no one was able to get very high salt rejection. And we said, let's do some simple modeling. So this is the work of mostly Cody Ritt, one of my PhD students. And we said, let's do some multi-color simulation, try to simulate this creation of this membrane and see what will be the performance limit of such membranes. So we created by layer, more like a vacuum filtration of a graphene or 2-dimensional nanosheets of different size. Again, here I showed you all the same size, but we'll have them different size. And 2 important parameters are the areal coverage. What's the coverage of the membrane of the area here by the graphene oxide without any overlap? Because once you overlap, you'll be, with some defects. And then the thickness. When you build it, what will be the thickness? And we have done this kind of Monte Carlo simulation to build this layer to any thickness that we need. And we challenged this by water molecules and sodium chloride. And we were able to measure the rejection and calculate the permeability and selectivity of these membranes. So I would like now to show you these results. It's a very loaded graph, and I will guide you step by step. This is the water source selectivity, which translates to this rejection. This is the water permeability coefficient. And we're starting from the right, we're starting to build the layer. And let's take an example of 50% coverage. If you build the layer, and then you build and build, then it becomes thicker, and the permeability will decrease. As you see, I'm moving here. And I stop building the layer when the permeability reach 1, because 1, it's already not energy efficient. And you see, if you have 50% coverage, the rejection can be at most 10%. Then I will show you for 75%, which is typically the best coverage that you can get in any laboratory study is 75%. And let's do the same thing, build the membrane, more and more layers. And as you get more layers, the permeability decreases because water needs to go through. And again, this layer can be 100 nanometer, and it can be very, very tortuous. You get a lot of long path of the water. So eventually we stop when the performance, the permeability dropped to about 1. And here we got rejection about between 70%, at most 80%, which is typical of what the best studies that reported on this topic. Then we went to 90%, which is very difficult. To get 90%, we need to have nanosheets of different size. And again, and we avoid any overlap. And no one was able to create anything like this 90% coverage in the laboratory. And we start again. We're starting to build the layer, so moving to the left, and it's become less permeable because the layer becomes thicker and thicker and thicker until we stop when the permeability went to 1. And you see here that we can get about 99.5% rejection. Now, this is good performance, but the performance of current polyamide membranes, the one that I spoke a lot about before, is much higher. So it means even when you go to 90% coverage, you will not be able to get this performance. So the question why? This is related to what we call framework defect that you cannot avoid by this method. It's a top-down approach. And I would like to describe this framework defect. This is just one layer that you see here. And imagine, and again, this is a framework defect. Imagine if you build more and more layers, this framework defect can be interconnected. And the only way to completely prevent, statistically prevent this framework defect, the membrane should be so thick such that the water permeability would be so low, so it's not practical. So framework defect control all of these, and this is the reason why this membrane, despite all these fancy papers in Nature, Science, etc., will not perform well. And again, from engineering, from my experience in many years in engineering, whenever I see anything that's published in Science and Nature that promise a new technology, I mean, usually it's good for the paper, but actually for the practical application, it never works. And this is just one example. So lastly, I would like to go to the last topic. And again, I want to be within the time frame that I was given about ion-ion selectivity. So this is the work mostly done by a postdoc, Razi Epstein, who is now a faculty at the Technion in Israel. And let's go to the current reverse osmosis membrane. So reverse osmosis membrane remove all the salt very well, I showed you, but they cannot distinguish between ions. The same thing with nanofiltration. Nanofiltration that you see here, again, this is rejection, this is nanofiltration, reverse osmosis, again, the pore becomes smaller. Nanofiltration can distinguish between monovalent and divalent. But what's about distinguishing between monovalent? I mean, I want some membrane that can distinguish between lithium and sodium, because then I can separate lithium, or between nitrate and chloride. So then I can let only nitrate, for example, go and get reject only nitrate and have chloride going through. And this is very difficult with current membranes, because current membranes, the mechanism is by either size exclusion or charge exclusion, which is mostly, again, RO and NF membrane. These mechanisms are good enough to get very high salt rejection, but to distinguish between similar ions, they cannot do it. And what I would like now to tell you how we can achieve it, and we can go to the biological channels. This is the potassium channel. Due to evolution of over 1 billion years, we developed the best selective membrane that you can ever think and imagine about. And this membrane has many features, but I can tell you that they can transfer potassium for every 1,000 or even more than 1,000 potassium, they will let only one sodium, sorry, for any 1,000 potassium ion, only for any 1,000 sodium ion that go through, only one potassium will go through. So selectivity of over 1,000 and more. And this is a very unique membrane. It's only the channel. The selectivity filter is only 12 angstrom, 1.2 nanometer, very, very thin. And it has many features that really dictate such selectivity, which I would like to describe here. First, they need to be in a size which is smaller than the hydrated radius, but in a way that you can have dehydration, and then you can have the ion going inside the channel. So smaller than the hydrated radius, but bigger than the dehydrated. And yes, and then you can have binding sites. So the way it works, once you dehydrate, again, dehydrate, you need to be small enough, but not too small, so it can get inside. And once it gets inside, you can have some binding site that compensate for this dehydration. When you dehydrate, the energy of the ion is high, and it feels very, very unstable. So immediately you need to find some functional groups to bind to stabilize it so it can move in the channel. So this is, so this is for the potassium. So you need to get inside and move through the channel. I think I confused you earlier. The potassium is the one that goes through and the sodium doesn't go through. So, and this channel transfer 1,000 potassium ion for each sodium ion. Then it can have very thin, because it's as I told you, it's 1.2 nanometer. If it's very thick, it will be very, very slow transport rate. And then the distance between these ions, between these functional groups that stabilize in such a way that the ions that come here pushing the ions that was before. So this is called the knockoff effect. And all these things, I mean, really making this a biological channel very, very selective. So of course, the goal is to mimic this channel. I will not show you the result that we were able to mimic it completely, but I would like to show you some directions. First, I would like to let you know that it's typical in this field to describe the selectivity or the transport through ions through this membrane by energy barriers. What will be the energy barrier for ion transport? For example, here I show you two energy barriers. The red one is high energy barrier, so it means none of the red molecules will be able to go because it needs to overcome a very high energy barrier. The blue one is a low energy barrier, which tell you that it's not enough to prevent the transport. And there are many causes for this energy barrier. They are written here, but again, they're beyond the scope of this lecture. So what I would like in the coming slide, just before concluding, to show you some results. Again, I want to see the timer. Okay, we are close to, John Go gave me 40 minutes. Here, we measure the energy barriers through ion exchange membrane, just to understand this transport phenomena. So we measure the transport of different cations and anions. This is, for example, nitrate, fluoride, bromide, and chloride. We measure the flux, and from the different temperature, and we use the Arrhenius type equation, the log of the Arrhenius type equation, and from the slope, we can calculate these energy barriers. And this is what you see here, the energy barrier for ion transport, fluoride, chloride, bromide, and nitrate. This is the direction of the hydration energy. Fluoride is the smallest ion, as the smallest ionic radius. So the water molecules bound very, very tightly, or in other words, there is high hydration energy. So it's very difficult for it to dehydrate and getting to the membrane, to the pore. And this is what we get, high energy barrier. And we see that it goes the same direction, except bromide and nitrate. The reason, again, is related to the polyatomic structure and not a spherical one, but I will not describe it in this lecture. So we see roughly it's in line of the hydration energy. We can also describe the pre-exponential factor, the A coefficient. You can think about it if there is no energy barrier, 0. So this will be just the flux or the diffusion. And we see we get the same trend. And the reason is that once the ion partition enter the pore, it needs to move through this ion exchange membrane and interact with the fixed oppositely charged groups. And because fluoride has the least dehydration, it has still water molecules around it, the interaction with the fixed group is weak, and this is why it can move faster inside the ion exchange membrane. So we see the same trend. So again, I wanted to show you here that there is some interaction with the fixed group, and the key is what we are working now is how to tailor this fixed group in such the right chemistry to mimic the potassium channel so we can really have the effect that I described earlier. So again, to summarize, I show you this evolution of next generation desalination membrane, all the way from ultra high flux to ultra high selectivity to what I think will be a lot of the future research on ion-ion selectivity. And the conclusions are that in terms of water permeability, we do not need high water permeability membrane. 2,3 LMH is good enough, and anyone who knows membrane knows that if you work with very high water permeability, you have a lot of problems, fouling and many other phenomena. Water salt selectivity, it's again, it's a major challenge because of defects, and I show you the examples of the graphene and biomimetic membranes. Ion-ion selectivity, I think it's an area of future research, and we need new approaches, and in my opinion, the new approaches will be sort of trying to mimic biological channels. And lastly, thin film composite melt polyamide membrane, despite the fact that they are 40 years old, they are still the gold standard. In my opinion, they will also be the gold standard for years to come. The only way, in my opinion, to have a better membrane is to have a bottom-up approach. All the approaches that I showed you are top-down. You take some building blocks and try to put them in some structure in a hope that you will get some good membranes. It's not the way to do it. The way to do it is to take some molecules and build from bottom up the membrane that you can design it. And again, this is the only way, in my opinion, to build the polyamide membrane, which, by the way, is good enough. We have many desalination plants. They solve water scarcity in many places in the world, but you can improve it to make it slightly more economical if you have high selectivity membrane. So lastly, I would like to conclude the funding agency and my group and some hello from Yale Universities. So thank you, and I'll be happy to answer any questions.